Hello everyone. In this lecture, I will give brief introduction to radiomics and how we can apply radiomics analysis to the neuro-oncological research. Brain tumors present a heterogeneous and complex nature, which makes their treatment and clinical decision-making difficult. Imaging such a heterogeneous tumors limits the predictive power of current MRI techniques. The improvements in the computational power of image analysis techniques have led to transforming medical images into mineable and high-dimensional data. The main idea is to link these quantitative features to tumor imaging characteristics, intertumoral heterogeneity, genetic characteristics, which is called radiogenomics, tumor phenotype, treatment outcome, to support clinical decision-making, personal treatment, prediction of prognosis, and so on. PyRadiomics is an open source Python package that was proposed by Harvard School of Medicine. This is a generic workflow of radiomics. As an example, we try to predict whether this tumor is benign or malignant based on multi-contrast MRI data. Then we must gather imaging data and delineate the tumor boundaries. To minimize any inter- and intra-patient variabilities, preprocessing steps are applied, such as spatial resampling and intensity normalization. Then we can extract radiomics consist of intensity, shape, and texture-based features. Since we are going to have a large number of features, feature selection and dimension reduction techniques are applied to select most predictive features. Based on our aim, statistical or machine learning approach could be employed. If you look at the steps more closely, to increase the reproducibility and generalizability of radiomics, several preprocessing techniques are applied before feature extraction. Discretization, spatial resampling, registration normalization are some of them. Filters are applied to enhance details, identify recurrent patterns, eliminate noise, and enhance edges, therefore increase the number of features. For instance, wavelet is used to enhance specific frequency components from the images. Laplacian of Gaussian is applied to enhance edges and fine details while suppressing the noise with Gaussian filtering. First-order intensity features compute common histogram descriptors such as mean, median, skewness, and kurtosis. Shape features are used to measure geometric properties of the tumors, such as perimeter, principal axis length, radius. 3D shape features can calculate how tumor shape deviates from an ideal sphere. As can be seen from the two tumor models, spherical one on the bottom has higher sphericity value, elongation, and flatness values, and lower spherical disproportion values, compared to more irregularly shaped tumor. Texture-based features are designed to quantify texture by analyzing the spatial relationship within the ROI. These features are computed not from the image itself, but from its matrix representation. Gray level co-occurrence matrix, GLCM, represents how often the intensity values co-occur in a specific direction. For instance, we have 4 and 1 intensity neighboring pixels at theta equals 0 direction. They occur 3 times within the ROI. GRLM counts the number of run length matrices within the same intensity values over a specific direction. In this image, we have 3 pixels connected at theta equals 0 direction. So our run length equals 1 for this combination. We calculated these for 2D case, but in PyRadiomics, it is calculated also 3D. And we have 3 more groups like gray level size of matrix, GLSCM, gray level dependence matrix, GLDM, neighborhood gray tone difference, NGTDM. After computation of all these classes, we are going to end up with a large number of features per patient. In order to prevent overfitting and make the models more interpretable, the most predictive features are selected. Supervised, unsupervised, and dimension reduction techniques are frequently used. Supervised techniques take the class label into account while eliminating the features. There are filter-based, wrapper-based, and embedded methods. Unsupervised techniques do not consider the class label like k-means clustering. Dimension reduction techniques convert features into new representation like PCA, which leads the lack of feature trap. Additionally, multi-level reduction techniques can be employed, like combination of these techniques, which can identify features more efficiently. After selecting the most powerful features, we can integrate the features with clinical or genetic data, or we can predict them. Depending on our vision, statistical test, classification algorithms, or survival analysis techniques could be applied to the radiomics. Radiomics has been increasingly applied in brain tumors to improve predictive accuracy of diagnosis, prognosis, and treatment outcome. The main strength of radiomics is to capture intertumoral heterogeneity by using texture analysis, as can be seen from the radiomics features computed from T1 and T2 weighted MRI. They are associated with different molecular pathways. Also in neuro-oncology, radiomics can be employed to differential diagnosis, WHO tumor grading, prognosis, and molecular alterations. Now we are done with the introduction part, and we will continue with a hands-on session for the radiomics. In this part of the course, we will work through a complete radiomics pipeline, which performs radiomics analysis using TCGA low-grade glioma dataset. These scans were already discussed, stripped and co-registered, and their tumor segmentation labels were produced by an automated hybrid generative discriminative method. TCGA data can be accessed from the link. Molecular biomarkers of brain tumors were integrated into typing, subtyping, and grading of major groups. Isostrate dehydrogenase, known as IDH mutation, and 1p19q codylation biomarkers have been identified as significant predictors for therapy and prognosis in low-grade gliomas. The current tutorial aims to construct a machine learning-based model for identifying molecular subsets of low-grade gliomas as IDH mutant and 1p19q non-codylated astrocytoma, IDH mutant and 1p19q codylated oligodendroglioma. A total of 49 low-grade glioma were included in the example. The sum of 80-20 radiomic features were extracted from T2-weighted MRI using PyRadiomics platform. Clinical information also simplified for better understanding and appended to radiomic feature files, T2 TCGA feature. The main workflow of this script includes feature extraction using PyRadiomics and loading the data, feature normalization, feature selection, then model training, statistical analysis, and also Kaplan-Meier and Cox proportional hazard survival analysis. The complete code can be found from the link, and any suggestions about pipeline you can email to this address. And we are going to first begin with loading required libraries. Before computation of features, preoperative TCGA data, low-grade glioma data, comprise of pretreatment scans and their associated segmentations for each patient. These segmentations include 3 glioma subregions as enhancing part of the tumor core, non-enhancing part of the tumor core, and edema. We generated merge versions from union of these subregions as a whole tumor and used for the computation of the radiomic features. Glioma 1 mm YAML file is an example parameter file for customization of feature extraction scheme, like resampling filters and specifying which features to be calculated. TC TCGA T2 directory file is a batch processing file which can where each row provides the patient ID, corresponding image, and mask locations. Additional columns are patient's clinical information, such as age, gender, survival data, etc. To run the tutorial, either you will need this TCGA features CSV file, and this is the file with the PyRadiomics output and appended to the directory file, and also can be generated from the following tab. So after that, we are going to load the data, and we are going to end up with 37 astrocytoma and 12 oligodendroglioma subjects, and we are going to have 18-20 features. Since Radiomic produces high-dimensional data, feature selection is crucial. We first remove features with low variance and drop highly correlated features with 75 correlation coefficient. And after that, we are gonna apply Lasso regression to identify most predictive subset of the features after splitting the data into train and test. And we are gonna apply Lasso regression to select the most important features. And for the model training, we are gonna apply machine learning models like SVM, KNN, Logistic regression, or AdaBoost to classify gliomas into astrocytoma and oligodendroglioma. We also evaluate their performance across 100 iterations to get the reliable accuracy, sensitivity, and specificity. Typically, logistic regression and SVM gave us the most stable performance in this example. Then the selected features from process of Lasso regression. We ranked the selection frequency over 100 iterations, and the selected features with the frequency above, like, let's say 75%, selected for the further statistical analysis. And now we will evaluate the most frequently selected features to see if they significantly differ between two subsets with one Whitney U test. So one Whitney U test help us to identify which features truly discriminate astrocytomas from oligodendrogliomas. And we visualize these results with the box plots and their associated p-value levels. For the survival analysis, we are going to apply Kaplan-Meier survival curve and also Cox regression. And with the Kaplan-Meier curve, we compare overall survival between subgroups like astrocytoma and oligodendroglioma, and there is no significant difference between their survival times. And then we apply Cox proportional hazard models to estimate how radiomic features, together with clinical features like age, gender, and tumor grade, influence the survival risk. And this will give us results like age only influence the survival of the low-grade gliomas and increases the risk. So to summarize, we start from MRI images, extract radiomic features using PyRadiomics toolbox, select most informative features, build machine learning models to predict molecular subtype, and finally relate those features to the survival outcomes. And this is the end-to-end pipeline, how radiogenomics bridges medical imaging, data science, and clinical decision making. Of course, this is an example of how radiomics can be applied to gliomas. Thank you for your attention.